You're gonna become a YouTuber. Ok. Bueno, muy bien. Muchas gracias a todos. Perdonen los errores de transmisión, pero bueno, muchas gracias a todos por su interés en el seminario. Gracias, bienvenidos. El día de hoy tenemos la fortuna de contar con la presentación del doctor Lindsay Shaw. Les voy a platicar un poco de él. El doctor Lindsay Shaw nació en Inglaterra. Él asistió a la Universidad de East Anglia en Norwich, Inglaterra, donde obtuvo su grado de licenciatura en el 99. Después realizó su doctorado en el laboratorio de Richard James en Nottingham, en la Universidad de Nottingham, en donde realizó estudios de estructura de colicinas en Escherichia coli. Después decidió hacer su doctorado en el laboratorio del famoso doctor Simon Foster en la Universidad de Sheffield, en Sheffield, Inglaterra, donde se enfocó en las proteasas secretadas por Staphylococcus aureus, de lo cual nos va a hablar el día de hoy. Posteriormente hizo también una estancia en el laboratorio del doctor Jan Potempan en la Universidad es difícil pronunciarla, Jagiellonian University en Cracovia, en Polonia. Posteriormente realizó un postdoctorado en la Universidad de Missouri en Columbia, en los Estados Unidos, en el laboratorio del Dr. George Stewart. Y posteriormente, en el 2007, se unió a la Facultad de la Universidad del Sur de Florida como un profesor asistente en el Departamento de Biología Celular, Microbiología y Biología Molecular, en donde ha sido director de estudios de graduados del 2009 al 2018 y fue promovido como profesor asociado y obtuvo su plaza en verano del 2012. Se convirtió en full professor, que es un profesor titular C en nuestro país, en el verano de 2016. Y actualmente tiene varias distinciones, como que es actualmente parte de la sección de una sección de estudio del NIH en la sección de patogénesis bacteriana, y además fue admitido en la Academia de Ciencias de los Estados Unidos en el 2019. Cabe mencionar que el doctor Shaw tomó su inspiración de estudiar microbiología cuando él tenía 13 años. A los 13 años tuvo una operación en su cadera, en la cual le tuvieron que implantar varios clavos en la cadera para corregir ese problema. Y ocurrió que se infectó de Staphylococcus aureus por una biopelícula. Entonces tuvo que tener demasiadas operaciones y tratamientos, pero bueno, fue algo que lo motivó al doctor para entrar a la microbiología y estudiar Staphylococcus aureus. Yo tuve la fortuna de conocerlo porque hice una estancia postdoctoral en su laboratorio por casi 2 años, de 2010 a 2012. Y bueno, aprendí bastantes cosas sobre esta interesante bacteria. Así es de que los voy a dejar con el doctor y por favor al final de la sesión les pido que guarden sus preguntas, las escriban en español, los invito a hacer preguntas. Recuerden que es muy importante hacer preguntas en una presentación de ciencia. Si no quieren abrir su micrófono, yo les puedo ayudar a traducir con mucho gusto. Okay, well, that was my Spanish introduction for you. So thank you very much for sharing your knowledge and giving us your time for this seminar. So the stage is yours.
Thank you. And I do what everyone says, so thank you for the kind introduction. I just have to presume that it was kind, but I know you're Antonio. So I got about, till, what was it, an hour, not quite 15 minutes? Is that how long?
Yeah,
I just don't want to go.
Yeah, that's fine. 1 hour. Take your time, don't worry.
Okay, I just don't want to be that guy that makes everyone stay here forever. But anyway, yes, I'm very pleased to accept this invite from former postdoc Antonio to talk to you guys today, and it's always nice to share our work. And so, yeah, I'm very thankful for you guys being here and interested in what I have to say. If you are at all interested in anything we say, you can follow us on Twitter, and then you can check out the lab too and see the people in it. But today I'm going to talk about mostly secreted proteases in staph, which is what I've worked on since my PhD. So I always start out my seminars with the same way, right? And so the idea of what motivation, it's the #1 indicator of success, I think, certainly in grad school, but academia. So why do I do what I do? So this is a normal human femur right here. And so you got a ball and cap joint. And so it's the case is a disease state where the cap can slip particularly in teenage boys before there's fusion between the two. So this is called a slipped femoral epiphysis, and you can move that cap back on, but it'll just fall off again, and eventually the femur grinds into the pelvis, and then the patient can't walk. And so this happened to me too on my left side. And so here they showed it in both hips. It can happen, but only happened in one. And so you have to roll the cap back on and then pin it. Here they show one. I had 3 pins around my cap to keep it in place. And 2 of the 3 pins that I had put in me at 13 had Staph aureus on them. So I lived with a Staph aureus biofilm infection from the age of 13 until they were able to take those pins out at the age of 18. So for 5 years, I lived with a staph infection, thankfully not methicillin resistant, so it was eminently treatable, but living with any staph infection is not a pleasant thing. So so the idea of hospitals and bugs and drugs was innately all I'd really kind of had to think about from the age of 13 as a teenager. So I tell people I didn't really have a choice about what it is that I was going to do with my life. So, high school and microbiology and Staph aureus, and that was it. So so that's how I got to where I am now. I'm just trying to advance the slides. My apologies, it's being problematic. There we go. So is it going to do that to me every time? No, it's not. So what do we do in my lab then? And we do 2 things broadly. The first is we essentially study control in the Staph aureus cell. And so this is a picture made for me by a former grad student, Andy Weiss, that shows central dogma, right? Transcription, translation, post-translation. So DNA makes RNA is controlled at the level of transcription by transcription factors, 2-component systems, alternative components of RNA polymerase. And we work on all of these factors. Converting RNA to proteins is usually controlled by regulatory RNAs. And I was dragged kicking and screaming into the regulatory RNA world by a former grad student and postdoc. And so we've worked on those too. And then even though protein's made, it's not necessarily active. It needs to be modified post-translationally, and proteolysis is actually the most common post-translational modification. Every single protein is subject to it, and it's not just simple degradation. So we study the post-translational modifications of proteins too. And then we do have more than just a sideline. For a long time, it used to be at least half of the work that we do, which is therapeutic development. And so not only working on a very drug-resistant bacteria like Staph, MRSA, developing therapeutics with chemists at USF and around the country and the world to treat the infections that they cause. So we technically do work with all the escape pathogens. Staph is my first love microbially, and it's my favorite organism, but each of the escape pathogens we work on in the lab, we do pathogenic studies with Beaumontii as well as Staph, and we use all 6 in our therapeutics work. And not just are they a fun acronym, but they're a really good collection of bacteria, right? You've got 2 gram positives, 4 gram negatives. The 2 gram positives are on the CDC serious list. The 4 gram negatives are on the urgent list. It's a similar kind of story with the WHO list as well. But beyond that, they're just a really good model of a collection of bugs to study the idea of colonization, transmission, and then resistance. And so they're a really interesting collection of bacteria, all causing an array of terrible diseases, and they're all hard to treat. So to us, they're a great group of bugs to work with. But as I said, Staph is really what I'm going to talk about today. And then about those diseases, I think is the next picture, because they're always gory pictures. Sorry, I'm not advancing again. It's been problematic. Here are the gory pictures. So the collection of escape pathogens can cause things from pneumonia to septic arthritis to endocarditis, burn infections, UTIs, skin infections. The list essentially goes on. And so they're a great collection of organisms to study for all kinds of reasons. And then, obviously we don't have antibiotics, right? The idea that we might be able to treat these things is rapidly receding, such that we find ourselves with this potential for a post-antibiotic era, of which I say is there no escape. Right, and so we find ourselves in some sort of biological arms race, and we are nowhere near this close to the bugs. We are so far behind them. We're kind of wheezing up one side of the hill and they're rolling down the other, because we just left so much of our time for discovery just gone to waste. So we really are very far behind them, such that we could only be 10 years away from a potential post-antibiotic era. And if that ends up being the case, at least in the case of Staph aureus, 80% of invasive Staph aureus infections, if not chronically serious, then fatal without intervention. So we could end up finding ourselves in a place, and this is a quote from Margaret Chan, who used to be the Director General of the World Health Organization, that if we reach the post-antibiotic era, then modern medicine, as we know it, is in effect over, because things like strep throat or scratch knee once again kill. So all these things that since the 40s or before that we've been lucky enough not to have to worry about being serious become very serious again. I mean, we take antibiotics when we go to the dentist, for goodness sake. Chemotherapy and transplants and serious things like that all require antibiotics and a lack of infection. So losing our ability to treat bacteria through antibiotics really causes a major problem for modern medicine. So that's just a brief introduction of the problem, and I think this audience understands the seriousness of bugs and drug resistance. And so I'll get into the work that we do in my lab, and I'll borrow a quote from a friend of mine. It says, really, I'm narrating the work that is performed by my trainees and students and postdocs. So all credit to them, and wherever possible, I have given that. So I'm going to tell you really about these secreted proteases that I've worked on since my PhD and how they regulate the process of infection in staph. So I was thinking about this last night. This is less and less true as the years go on, but I've for years said that I find secreted proteases very confusing, and that's what this sign is meant to suggest, right? Because I myself have published papers that say proteases are very important for Staph aureus infection. They are not important for Staph aureus infection, and then they might be important for Staph aureus infection. So I'm supposed to be someone who knows about proteases, and I'm not even sure if they're important for infection. And this certainly was the case as I was starting my lab, finishing my postdoc. And the reason that we don't really, or didn't really know, I think we do now, is that there are 10 of these things. So Staph aureus secretes a myriad of factors into the host during infection to cause infection in every single part of the body. And so 10 of these factors are proteases, 10 different secreted proteases. And the problem is that they have overlapping specificity, right? And so you take one of them away and the others cover for it in terms of function. And so that was always the problem with the early studies, is we delete one and ask what happens, a basic mutation and phenotype study. And often the answer was not much happened when you deleted that gene. So that was the confusion part. So as I was telling Antonio before we started, I started my lab not on secreted proteases. I tried to get away from them and started the lab on something completely different, but kept getting dragged back to them. And so these are at least the secreted proteases and friends, and it's not really important who's who. But amongst these genes, 10 of them are the secreted proteases. And so I kind of had this idea when I started the lab that, well, we need to figure out if they're important in infection or not. It's this idea that they might, they're not, maybe they are, who knows. So we took this approach where we just deleted them. We took away all 10 secreted proteases of Staph aureus. So we made a 10 gene deletion mutant, or rather with our colleague, Alex Horswell, who's now at Denver, made the mutant. And so we made this strain and said, what happens now? If we take away all 10, then if there's a phenotype, at least we can show they're important. And if there's not, then we can walk away. And this was all work done by, or mostly done by a very talented grad student, Stacy Kohler, who left the lab now 10 years ago. It's a long time. So very simply, we're going to have mutant and wild type in the same kind of color. When you delete 10 genes, complementation of 10 genes in staph is not possible, but we've genome sequenced the whole strain and there are no additional mutations. So we know that everything we're seeing is a consequence of those mutations and nothing else. And so when we grow these strains, wild type or mutant, just in rich media, there's no phenotype. And there shouldn't be, right? Because these are accessory factors secreted into the host to facilitate infection. So in TSB, you shouldn't see an effect. We tried a media that consists solely of peptides or solely of peptides to a point. So here the bugs are burning the carbon source in the media, and then they're kind of using up peptides during kind of a starvation phase over time. And even though both decline, there's maybe is a half a log to a log difference. And so the mutant is a little impaired in a peptide-based media, which makes sense, right? A protease would cleave other proteins to generate peptides, which then the bug could eat for nutrition purposes. So more of a relevant phenotype, but that's got nothing to do with virulence. And so we moved on quite quickly and said, what about something like serum? So we grew the wild type and the mutant in serum to see if it had an issue in terms of defense. And sure enough, at least in the early adaptation phases, the mutant really struggled for growth in serum. And at least early on, students would always do the serum assay. And I would say, well, that's great, but what does it mean? How is serum killing the bugs? Because it's a gram positive, right? And so the MAC, the membrane attack complex, doesn't work against gram positives, and that's in serum. So what's in serum that's causing this effect? And the answer potentially is antimicrobial peptides, right? These things are produced as an ancient part of the immune system. They're present in serum. And so are these antimicrobial peptides that attack invading pathogens the reason for this defect? And sure enough, that was the case. And so here are 3 different human antimicrobial peptides with our wild type and mutant looking at percentage over time of survivability. And the mutant just can't deal with being attacked by these antimicrobial peptides, particularly as the concentrations increase, whereas in certain cases, the parent is doing just fine. And that makes sense, right? That you secrete a protease into the host and there's an antimicrobial peptide comes along and it's just going to chomp it up as a form of protection and survival. So that kind of made sense. And there had been one report that suggested this might be the case, but we're showing on a kind of in vivo level in the bacterium that the proteases are important for this. But we're not full of serum, so our next step was to move into blood. And so we thought, well, now, okay, do we see a difference in survivability of the mutant in blood? And so sure enough, we do. And so at least a 4-fold decrease, which is bigger phenotype than our serum, we've got a big defect for survival of the mutant in whole human blood. And so we're again asking, why is this the case? And so what's the difference between serum and blood? Well, many things, but we've now got cell-mediated immunity, right? And so is it because our mutant strain is just not as well adapted to dealing with the host in terms of cell-mediated immunity? And sure enough, that's the case. And I plug Antonio here because he did these experiments. I remember when we got the reviews back on this paper and Stacy had already left, and it was a case of someone needs to do this. And Antonio was more than happy to step up and do these great experiments. Really made the difference getting this thing accepted. So what Antonio did was he flagged each of these strains with GFP, and then we did an infection in the whole human blood. We isolated the cells and then kind of did a gradient type thing to separate the cells out and ask the question, is our mutant more likely to be resident inside leukocytes? And yes is the answer. It cannot resist uptake and or it cannot facilitate escape. So it's impaired for survival because it just can't deal with the cell-mediated immunity aspects of things. And so that made a lot of sense in the end. But ultimately, right, that's now moving from in vitro to ex vivo, but what about in vivo? Things that actually affect infection. And so here, what we do is we tail vein inject the mice with Staph aureus. It gets into the blood, it disseminates out to the organs, and then you get these abscesses in the organs. And this is not obviously a mouse, but it's just demonstrative. And so we get this dissemination to the organs. And so we said, oh, now 10 gene deletion, what's going on? And now we're getting some real phenotypes. We're getting at least a log, if not multiple log decreases in survivability of the mutant. And so we're looking at different organs here. The mutant, if it makes it to and disseminates to these organs, it just does not survive as well over the course of a 7-day infection. And so this was more like it. And this is actually kind of a big deal because in Staph aureus, there are so many virulence factors that single virulence factor deletions don't typically give you a big change. So we're talking multiple logs for all the proteases. So to us, that means something. This is a big defect. So one of the other things that you get with this model is that you can also measure survivability. And so when you do the tail vein injection over the course of a few days, some mice are going to die. And so for those mice, we don't include them in the dissemination pool. And so we have to do a lot of mice for this, but we're able to measure also survivability. And then we did it and we're like, oh, great, there's a massive phenotype. This is wonderful. But it was actually the 10 gene deletion strain that was killing all the mice. Right? This is a huge difference. The wild type is not killing that many mice, maybe 20%. More than 50% of mice infected with our 10 gene deletion are dying. Those of you that have made mutants and work in pathogenesis, you don't delete the gene and expect to make the bug more virulent. So we end up making this thing into a hyper-virulent killer. And so it was all going so well. We were like, why does this happen? And for a long time, I just said, I'm done with this project. I hate it. I told you I don't like proteases. Why are we still working on them? I told Stacy she'd mix the strains up. I said, go back and do it again and do it again. And so we're talking hundreds of mice. And that just was the nature of things. The way we did these experiments, we had to do a lot of mice. And these numbers have since been added to by other students in the lab, one of whom Jessica's on the call too. So, we've done this many times, and this is the kind of accumulated work of 3 different people over many years. This is the phenotype, and it's very robust. And so, as I said, I thought initially we mixed it up. This didn't make any sense. And so we stopped working on the project for a while, and then eventually we came back to it. And so I came back to this picture, right? And so here's Staph aureus, and here are the myriad of things, and this is most but not all of the things that are secreted into the host. And this is a remake by a former postdoc, Ronan, of a classic picture of Staph aureus virulence factors, and here are the proteases. And so we said, what about this scenario? And so we said, well, here are all the different virulence factors that Staph aureus makes. And here are the proteases. And I always try to put the little logo of wherever I'm talking on. So hopefully I think I picked the right one for you guys. So here is the proteases of Staph aureus, chompy, 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 right? And so proteases cleave other proteins. What if the proteases of Staph were keeping the toxins it makes in check? So one of its jobs is to cleave self-proteins to stop them getting out of hand. We just took away potentially guardians of the cell. We took away all the proteases, like so. And so now all the toxins become hyperstable. This now becomes, the strain now becomes like a teenager whose parents went away for the weekend. It goes nuts because it's got all these toxins and no one to keep kind of charge of it, to be in control. The proteases are gone. So is that why it's a hypervirulent killer? And actually, this was one of those times where science did what it's supposed to. It worked. And so sure enough, our mutant strain, I'm showing ratios of many well-known virulence factors in Staph aureus, and they're just way more abundant in our 10 gene deletion protease mutant strain than they are in our wild type. So that's what we've done. The proteases were keeping the virulence factors in check. You take away the proteases, the toxins go through the roof. Then when you inject it into a mouse, you create this massive toxin storm and you kill the hell out of many of them, which is unfortunate, but we learned, I think, something very important. And so, yeah, as I said, that was one of those rare moments that things actually worked. So that work was published some years ago now, and there's Antonio. He was on that paper, and so this was kind of founded my second act of working on proteases and many more things to come. So, so from there now it becomes kind of an interesting thing, right? It's a whodunit, right? Who is responsible for this hypervirulence? Because do we have to delete all 10 proteases, or is it just a couple of them are required for this outcome? And then, you know, it's not the loss of the protease, right? It's the overabundance of toxins that are causing the death. So how can we unpick this mechanism to figure out what's going on? And so that's what we started going about and doing. And so I don't want to get into the semantics of it, but our 10 gene deletion of proteases really is just 5 different genetic loci, and these are those. And that's because 6 of them are contained in this one spot over here. And we consider these, for at least these purposes, to be one enzyme, even though it's 6. So as I said, if you try to do the math of 10 gene deletions and combinations, it doesn't work. That's because we consider it a 5 loci deletion. Like, if you really care, we can talk about that later. So that's our complete null strain. And so then we said, well, what are every possible combination of protease mutant you can make in Staph aureus? And so that's what these are, right? We can begin to, quote, add back individual proteases to what we call our quadruples, and then add back 2 to our triples, and add back 3, and we get our double mutants. So this was all work done by a wonderful grad student, Brittany, who left the lab, I guess, about a year ago now. She's at Vanderbilt as a postdoc with Jim Cassatt. She did some amazing work on this project. So she made every single one of these strains, and those of you who've made Staph aureus mutants know that this is an awful thing to give a student, but Brittany did it with real aplomb. It's amazing. So we start by testing the virulence. So we're going to go down the list. And so we're going to ask the question, do we maintain the hypervirulence of this strain as we begin to add back proteases? So we're going to measure the quads first. And so I use the phrase adding back. So in these strains now, we're adding back 1 protease at a time. So in here, we've added, and unless you're an expert in Staph aureus proteases, it doesn't really matter, but we've added what's called the V8 protease. So all the other ones are missing. Here's the wild type, here's the mutant. Green is that quadruple, not quintuple mutant, and it's tracking hypervirulent. And so this addition of this one alone did not give us a wild type phenotype. Same thing here and here, right? The green line tracks the red line, and this one even more so, we're getting hypervirulent still. So the addition back of these proteases is not giving us restoration to wild type. And I think I know why this is occurring, because I think you get the hypervirulent phenotype because of loss of some of these guys, and then you've given the bug these back, and these I think actually have a very important role in infection. So I think that's what's going on here. And I don't want to get sidetracked with that, but I'm happy to talk with people if they want to about that afterwards. Interestingly, there are 5 possible quad mutations to make. When you add back either oral isin or staphylococcus A, now you begin to get more of a wild type phenotype, right? So the green line is not tracking with the red line anymore, it's tracking with the blue line. So we give it back a single protease, and now we're not seeing hypervirulence anymore. We're still seeing some virulence, right? We're still killing mice, but we're not doing it in the way that the parent was. So next we tested the triples, and this is a bunch of negative data, right? Now we're beginning to add back 2 enzymes. And so there are many combinations of triples, and many of them are not hypervirulent anymore. You see the green line track with the blue line. And so this doesn't really give us insight into our hypervirulent phenotype, but some of the triples still maintain hypervirulence. And so in this example, we've got the green line tracking with the red line, green with the red, green with the red. And so, all right, that's a ton of graphs. What's the common denominator here? And that's this, right? Every time we see hypervirulence, it's because these two enzymes alone, these two enzymes together are missing from the bug. Doesn't seem to matter which other protease you give them, it's just these two together. And that, if we go back here, is the same ones here that when you begin to take it away from the total knockout, you start to interfere with virulence. So we kind of began to think, well, maybe it's just these two. Maybe it's not all 10 that need to go missing. Maybe it's just these two, aureus and staphopain A. So we make that double mutant, and sure enough, that's exactly what happens. So here is that double mutant, and it tracks perfectly with the complete protease null. So the deletion of aureus and staphopain together is necessary and sufficient for hypervirulence. You don't need anything else. It's these two enzymes alone that give you that outcome, which I think is pretty cool. It really begins to narrow down the phenotype. So that at least solves one part of the problem. Who's giving this hypervirulence, or at least how is it occurring? And that becomes the next question. Because as I said, it's not the loss of a protease, it's the proteases are cleaving other toxins. So when they go missing, toxins become more stable. So which toxins are giving us this? Is it just any of them, all of them? Is it specific? So now we use some proteomics to answer this question. And so if you kind of bear with me for a moment, in our wild type strain, there are no proteases. Oh, sorry, no, completely wrong. In our wild type strain, all the proteases are present, right? They're all there naturally as they should be. And so they're kind of wandering around chomping up virulence factors. So we consider our wild type secretome to have the least amount of virulence factors. Still plenty for a regular infection, but the least amount intended. In our completely null strain, there are no proteases. So all these virulence factors are abundant. So we have tons and tons of virulence factors in our null strain. And here's that double mutant. So it's not going to have all of the virulence factors of the complete null, but it's going to have more than the wild type. So what we were looking at is a secretome of each of these three different strains, and where these two overlap, we wanted to see toxins that were overrepresented in our two hypervirulent strains that weren't overrepresented in the wild type. And so Andy Frey, a postdoc that's on this call, did this work with Brittany, and he figured out which it was. And so here are all our various different important virulence factors in our protease null and our double mutant, and their fold change over wild type. And as you can see, we're getting, in this case, really big fold changes, 50 or 60-fold increase in stability of certain virulence factors over the wild type. And so this gives us a bunch of candidates that could be important for this hypervirulent phenotype. So we ran with 4 different genes as part of this list. A couple of these things here, this guy and this guy, they're kind of surface associated, not classic virulence factors. So we were looking for something a bit more obvious. So Leuka is a very well-known leukocidin. The enterotoxin K is also a very well-known virulence factor, but also low abundance. So we picked that one as a low abundance one. This is a well-known and kind of still like 10-fold overabundant. Spin had recently been discovered as a secreted virulence factor, so we kind of picked it. And then this one is we knew nothing about. Actually, it was new to us, a completely unstudied but secreted factor in Staph aureus that is really super abundant in our mutant strains. So what we did was we took our double mutant that's hypervirulent and knocked out these guys along with it to see if we could temper that hypervirulent phenotype. And again, it was one of those cases where it actually worked. So here is the hypervirulent double mutant right there. And then we also knock out that leukocidin with it. And now we're really beginning to slow down and stop that hypervirulent phenotype. We're almost, it got to the last day where we did get a little bit, but it still tracks with the wild type, not the hypervirulent double mutant. And then that completely unknown, never before studied virulence factor, you knock that out alongside the double protease knockout, completely lose the hypervirulent phenotype. We're not seeing this hypervirulent phenotype, we're much more like wild type. So this was fantastic. Never before studied virulence factor in Staph aureus, and you take it away from a hypervirulent strain and the hypervirulence goes away. So we're really excited about 0964 and what it does. This isn't true of all virulence factors, right? And so there's that enzyme spin. It's pretty hyperabundant, but it's kind of sort of in between. So necessary, but not sufficient, maybe. And then enterotoxin K has no effect on the phenotype. It still ends up being hypervirulent. And that kind of makes sense because it was low abundance. So we kind of figured it out. We figured out what are the 2 proteases important for this phenotype. And then we discovered which virulence factors give us the hypervirulence. And to boot, we discovered an entirely new virulence factor as part of the process. So that work was actually published earlier this year in NBio, and we were quite pleased it was an editor's pick. And so many years of work by a lot of people, but I think we figured out some kind of useful stuff, and we now have many more questions to ask about our collection of protease mutants and also the virulence factors too. So so that kind of gave us our next step, or our next cue of what else do these proteases do, right? Because they're produced by the bacterium, and so essentially everything I just showed you was its role on cleaving self-proteins. But these things are produced from the cell and away they go, right? They can diffuse through infection. And so they drift off, as shown here, into the host during infection. So the next part is that, well, what are the targets attacking host? We've looked at attacking or degrading self. Now what about attacking host to facilitate infection? And so that was kind of the next part of what we wanted to do in this work. And so to do that, we used proteomics to understand substrates of the proteases targeting the host. So conventional proteomics would just take a bunch of proteins, trypsinize them, run them through the mass spec, and you ID them, and that's what you get. But really, that's kind of a sledgehammer approach for what we were wanting to do. We wanted to kind of more of a scalpel, not a chainsaw type thing. And so this is work by Andy Frey, a postdoc in the lab, who's on the call. And so he did an awful lot of work in this study of looking for substrates identified from the host. So what we used was a process called N-terminomics. And so N-terminomics allows us to specifically identify cleavages within proteins using proteomic techniques. And as I said, it's much more of a scalpel than a chainsaw. So what we did was we took serum proteins from humans, human serum proteins, and we took them alone or incubated with V8, which is one of the staph proteases, SSPA. And so we either don't treat or we treat. And so you can imagine if you look at the protein abundance, and this is just a cartoon, if you treat with the protease, you're going to get cleavage events in this pool of proteins that you don't get occurring here. So new N-termini will be generated by treatment with this protease that don't occur here. So what you do next then is you block those N-termini. And so we're going to have different N-termini in the untreated versus the treated. We'll have more in the treated because we have cleavage. And so we block those N-termini. Then you trypsinize because these fragments are way too big to stick on the mass spec. So we've got to get them small so they can stick on the mass spec. And then we use this polymer that takes out everything that doesn't have a blocked N-termini because those don't represent our protease cleavage events. They represent trypsin cleavage events. And so we now can selectively block our cleavage events, and they're differentially labeled to heavy and light labeled. So we use the polymer, so we get rid of the non-cleave factors, and then we can pool them together because, as I said, they're differentially labeled, so we can tell the mass spec and the programming to figure out who's who. So yeah, stick them on the mass spec, run and see what we get. And I love this click, and I suspect Andy does not love this click because this click is in 1 year of his life. A year later, the whole thing worked. And so in that click, Andy figured out what took a year to make this process work. And so when we take away anything that's not blocked on the N-terminus, then we look for only unique peptides present in 3 replicates, and then unique to our treated sample only, we get 191 possible cleavage events. And immediately what we did was we looked at the N-termini of these, and we realized right away that it had worked. And that's because the V8 protease cleaves very specifically after glutamic acid residues. And so in our untreated N-termini that we got, it was mixed across all different possible polar and non-polar amino acids. But in the treated with V8, almost all were with a glutamic acid cleavage, which is its known preference. And so already this says, okay, that's not pure chance, this has worked. But of course, we have to validate that these cleavage events actually occur. And so the next thing is that Andy creates these maps of the potential target proteins, and then he can do digests of, in silico digest with trypsin and then with V8 protease, and kind of figure out what he thinks will be the banding pattern that we would receive if we were to Western blot for these things. And so there's the full length of serotransferrin, which is one of the targets. And here are the various different things we think V8 would give us as cleavage patterns. So Andy then incubates serotransferrin with V8 at increasing concentrations, and we begin to get a degradation pattern appear. And then you can line it up. And so we begin to see the various different size fragments that we expect. So there's the 49 kilodalton one, and then we have one that should be at 40, and there it is, and then one at 25 too. And so we begin to get predictable cleavage patterns from what we predict occurring. And so the mass spec tells us this happens, and then we do it through straight biochemistry and Western blot, and we get the same outcome. And so again, this is validation. And so I'll show you a couple more. So here's alpha-2 macroglobulin, same thing. Here's the banding patterns we expect, and there are the products. Same thing again with SerpinC1. There's the expected product, and it happens. And Andy did this, I think, 10 different times. And so we, for the review of this, we did it a bunch of times, and it worked. And so our mass spec approach to understanding targets identifies actual targets of this enzyme. For validation further, because we all know how journals can be about validation, we then bought the protein. So now we're buying alpha-2 macroglobulin. So the previous examples, these were in serum. So we just added the protease to serum again and see if we could see what we want. Here we bought the proteins treated with the V8 protease. we get cleavage bands that we expect, and then we cut those out, we mass spec them, and they're the products that we expect them to be. So that happened for alpha-2 macroglobulin. This is from the complement C3 factor, and same thing, we get the cleavage bands that we might expect, we cut them out, send them for mass spec, and it fits with what we've seen from N-terminomics and Western blot. So we get validation of these things as targets from human samples. And so then Andy took all the various different, I think 80 some cleavage events for different things that we'd seen and puts them into ingenuity pathway analysis. And this kind of pops out. These are the various, everything in orange is cleaved by the V8 protease. And so these are all the different factors that are cleaved. And the fun thing is that some of these are already known targets of staph proteases. People will take one human factor and one protease and put them together biochemically and say, oh, look, we've got cleavage. We en masse in one experiment identified 80 plus of these things, kind of an unbiased global approach. So some of these, so we validate in a global way what people have done in a one at a time type sequential approach. When these break down, we find protease inhibitors are very much targeted by the staph proteases or the V8 protease. And this makes sense, right? These inhibitors exist for a couple of reasons. One would be to inhibit the action of invading proteases. What we find is that instead of the bait loops that the protease is supposed to get stuck on, the staph enzyme will go and cleave around the back and destroy the inhibitor. And so it's completely just going about and evading the effect of this inhibitor. The other effect is if you destroy protease inhibitors, now the inhibitor cannot inhibit host proteases, right? And we are full of proteases. You let those things go wild, you start getting dysregulation and destruction of host tissue. And so essentially the V8 protease can dysregulate protease activity of the host and cause damage, which can cause tissue destruction to its benefit. Complement's an obvious one, right? You start to destroy complement factors, then we've got immune functioning that's not working properly, signaling and those kinds of things that stop mechanisms that are designed to kill invading bacteria. And then we also kind of understand that bacteria like to engage the clotting cascade, which itself is dependent on proteolysis. And they'll do this again because they can either bind to those factors and cloak themselves with them or chop them up and eat them and all kinds of things. So these are sort of things that we thought might be the case anyway, and they turn out to be true targets of them. And then phenotypically, this also ended up being important too, because this whole experiment was just serum proteins and a protease, and we get outcome. But does that make a difference to the cell? So here what we did was we took the wild type Staph aureus, or one with this protease deleted or complemented back, and just stuck it in human serum and said, can it survive? And then over the course of 2 and then 3 and then 4, 5 and 24 hours, we do see that the mutant really doesn't like to survive. They cannot survive particularly well in serum compared to the wild type and the complement. And so those cleavage events that we'd identified through various different manners actually influence survival of the bacteria. And that's important, right? Remember this picture, there are 10 proteases, right? So you take one away, you don't always see a phenotype. Here, we've identified what we think are important targets, and we do see a phenotype. So it's phenotypically relevant and linked to the data we just found. So we think this kind of tied a nice bow on it in a very simple experiment that said, yeah, the findings that we just presented are actually important. And so we just published that one actually earlier this year too in Cell Reports, and we were pretty pleased with that. I think it's a pretty nice paper and one of the first use of N-terminomics to study a bacterial protease attacking a host. Andy, and this is just a very quick slide of some of the things that we're doing after that Andy's been doing with us. So we're really interested in using proteomics now in terms of studying how infection occurs inside the host. And so usually if you're trying to study things at a proteomic level and during infection, you've got all kinds of host factors that are confounding, which are way higher levels than the bacterial, so it's very hard to detect. So using much more targeted methods, Andy designed a library of potential or known triptic peptides from individual virulence factors, and these are all the different bipartite toxins and alpha toxin. And so kind of peptides that would reliably from pure culture elute at a particular retention time. And then looking at the MS2 ions that come off of that and being able to reliably detect them for abundance at the window we expect them to. So this is kind of a targeted method to doing mass spec. You kind of have to know what you're looking for rather than the unbiased approach. And so by taking this library of virulence factors, we then applied it, or Andy did, to an infected host. And so now we've got a mouse infected with Staph aureus. And so we extract the proteins from the kidneys, and then we use the same retention times and looking for those MS2 ions at the various different virulence factors that we've identified. And we can see them because now we're kind of clearing, it becomes, rather than looking for a needle in a haystack, it's the complete other way of looking through the telescope. So now only we're looking for the virulence factor, and so we can see it very easily. And so we get nice MS2 quantification of each of these different virulence factors from infected kidneys as well as from pure culture. And so we can now begin to kind of quantify these compared to pure culture across the organs during infection over time. And And just as one simple example of something that Andy did with that is this is looking at the staphylococcal complement inhibitor skin. And just looking at his quantitation over 5 different organs in the mouse so I was just showing kidneys here. This is the same infection, but 5 different organs stacking on top of each other, the MS2 ions. And we can see that skin is way overrepresented when you control for CFU in the lungs and the spleen, maybe some in the liver, but not so much in the kidneys and the heart. And so, this method now tells us we can begin to look at the abundance of different virulence factors within a host during infection in different niches. And then if you begin to think about it, you could do it every day and look at it longitudinally and really begin to zoom in and examine how an infection occurs and progresses from the bacteria. So kind of some of our next generation type aspect approaches to studying virulence are pretty cool. But to kind of bring it back to something that many in the audience are interested in, and that's kind of genetics. That's kind of what I'll spend the last bit of time on. But to kind of just finish that mass spec stuff, right? We just did the V8 protease in serum, and that was, it was a really important, I think, really good study. There are 9 other enzymes. We could look at all of them in serum. We could look at them in leukocyte proteomes or from organs, all kinds of different things, right? So the power of N-terminomics to witness cleavage by attacking the host is completely almost endless, and is still Andy has done a lot of that work already, and so we've got a lot more we can do. But as I said, I'm going to segue now to regulation, because of these 10 proteases, they're controlled from 4 different promoters, right? There's 1 here, there's 2 of them here, 1 here, and then 6 here. So 4 different loci control the production of these 10 proteases, and I'm keeping an eye on the time too. So we and others over the years have contributed to the knowledge of protease regulation in Staph aureus, and this is just meant to be kind of an idea of what we sort of know or think we know about how proteases are regulated. But we think this is kind of the tip of the iceberg, because I think what I've showed you so far hopefully suggests that secreted proteases are really important to controlling how infection progresses. And so this is a figure that I love that really kind of explains what we do. It was made by a former grad student, Andy Weiss, in the lab. And so we believe that toxins and proteases exist in this balance, right? And so we think that at certain times, there is a need for toxin or toxins, and for them to become overrepresented in a population or niche, the proteases have to be tamed, they have to be tamped down. But similarly, there are environments where we really don't want toxin or toxins, and so we have to have a way to tighten up the proteases, right? And so we get this yin and yang, this balancing. They can stay in stasis, or we can kind of dysregulate proteases to get more toxins, and vice versa. And it's not just en masse as we're showing here, right? We could titer out individual proteases to titer in individual toxins, right? They could really fine-tune infection by selectively controlling the production of key virulence factors. And that's why we think that they're so important to the infectious process. So understanding how they are regulated in turn affects how toxins are produced, and thus how infection occurs. And so to us, this becomes very important. And so this is a new kind of fun little technique discovered by that same grad student, Andy Weiss. And so he developed this in his postdoc lab with Eric Scar, and it's something that we've worked on with them and helped do some validation work for them. And it's kind of a fun screen. So in Staph aureus, we can use transducing phage. So we can use a phage to pick up a gene deletion marker, and we can move it to another strain. And genetics in Staph isn't easy, but transduction is just absolutely wonderful. It's the best thing that ever happened to Staph genetics, and it's been around a long time. We're so fortunate to be able to do it. So what that means is that we can create, say, a fluorescent plasmid. We can fuse our promoter of interest, go back here, take any one of these promoters and stick it to something like luciferase, and then en masse transduce that in a 96-well plate into any mutant that we want. And we have the complete Staph aureus knockout library, the Nebraska transposon mutant library. So 2,000 and some genes, we can just drop our fusion into every one of these and then read the luminescence and see how things are regulated. And there's an example, this is some data from a student of mine, Jessica, who's on the call, who studied aureus, and she's done plates and plates and plates of these things, but this is just a quick heat map she gave me last night to show the kind of data that you get. And so you can track over every kind of 15 minutes or whatever you want, a time window. And you can see in the wild type aureus, the expression peaks at around 3 hours, but in Rex, that expression goes away. It probably goes up a lot in RJR and UVRC. So you can get just tons of data by using this method. So we're really interested in studying protease regulation. And this is one of the newer approaches that Andy and Eric developed that they've been kind enough to share with us. But in a more kind of cohesive way of doing this, we did a screen where we wanted to look at protease activity. And this is actually a picture from my own thesis. So here's the activity of secreted proteases of a wild type and mutant, and you can run them on a cymogram and see what happens. And so this is work done by Brittany again, a wonderful grad student, and she used the Nebraska transposon mutant library to look for regulators of protease activity through zymograms. And so here is essentially that, and I want to plug this. This was all facilitated by a paper that Antonio worked on with Ernesto in my lab and then beyond, which was to in silico identify all the regulators in Staph aureus. And so Antonio did this wonderful study, and we had this great list of all the known regulators, which has actually become quite a big deal. A lot of people reference that paper, which I'm sure you know about, Antonio. So anyway, we took every single one of the mutants for those regulators that we'd figured out, and then looked at their protease activity using these gels, and then using the image J, and we ranked them. And so we get this big list of all these different possible regulators of proteases. And so we get a couple of really interesting things. I'm going to give you kind of more general things before I close with an interesting story on one of those regulators. But we can then learn, well, we can take one of those mutants and zoom in and say, all right, which of the protease loci is affected? And sometimes they're all affected in one way. Other times we get regulators where it's only impacting one, but not the others. And then we get something like this guy, XDRA, which is my new favorite regulator, which I don't think I'll have time to tell you about today, which just has profound impacts on lots of different proteases. We can then look at those individual regulators and say, all right, well, are they doing it themselves or through known regulators of protease activity? And sometimes they don't seem to affect any of the regulators that are known at all, so it's probably through their own activity. Others, like this guy, potentially working through SAE and maybe SARS. And then XDRA, again, is just my favorite because it affects everything. The XDRA mutant truly does seem to do everything. I don't know that I'll get to tell you about that today, unfortunately. So Brittany essentially took all of that data plus a whole bunch of other stuff and made this regulation map. And so this is what we call the kind of framework for protease regulation. Now we know that this is absolutely not going to be perfect every time and work exactly the way we show it, but it gives us a testable map, a way to begin to think about proteases here and how they are regulated and how they cross-regulate each other as well. And so this gives our testable map of protease regulation. And that was something we published a couple of years ago in EMSphere as part of Brittany's thesis. And not one of the things from this, but one of the kind of really important regulators that we have kind of brought out of this and other screens was something that Stephanie worked on, who was having a chat in Spanish with Antonio before this all started. And so we discovered this thing called MROQ. And so this ends up being a regulator, a membrane regulator of quorum sensing. It controls AGR. So here's a predicted structure of MROQ, and we know it sits in the membrane because we tagged it with HIS, and it's only present in membrane fractions. And so MROQ started, and we published this in I&I a couple of years ago, and we were fortunate to have this as an editor's pick. So we started this by a screen where we were looking for protease regulators, right? And an MROQ mutant on a casein agar plate has way less secreted protease activity that complements back. And so we saw that and we thought, well, is it just protease? And so we tested hemolysin, and same thing, you get a reduction in hemolysin production by an MROQ mutant that complements back. So this was kind of cool. So we said, what about other virulence factors? So here is a PVL leukocidin in the mutant. It goes away, which complements back. And then we tested a surface protein, which are regulated in the opposite way to virulence factors, and we get the opposite. The mutant has tons of the surface protein, which we set the threshold low for this, so we wouldn't get a burnout in the mutant. We don't see as much of it in the wild type or the complement, although if you leave it long enough, it does begin to appear, but this will burn out. And so if you know anything about staph regulation, this just screams AGR regulation all the way. These phenotypes we're showing are classic AGR regulation. So here's the AGR system of Staph aureus, the major quorum sensing system. And I won't spend time talking about it other than here's the quorum sensing module, and here is a two-component system that senses that and then gives you outcome, which is through this thing called RNA3. And the first thing we noticed was that MROQ, our target gene, was actually really close to AGR in terms of where it's located on the chromosome. We tested the expression of the AGR operon, so there are two main promoters here and here. And in our MROQ mutant, they just flatlined. They were way down. So we know that, yeah, absolutely, this thing is affecting AGR activity. So we said, okay, is this occurring directly at AGR, or is it working through known regulators of AGR? So these are a bunch of known regulators of AGR, and some of them are changed, but most of them, if they are changed, are in the wrong direction, so they can't explain what we're seeing. Some are changed in the right direction, but we're talking a couple fold, and we're seeing hundreds of fold difference. So it doesn't look like it's a direct regulation issue. So we did RNA-seq, as we do in our lab a lot. And so we did RNA-seq of the wild type and the MROQ mutant. We did RNA-seq of the wild type and the AGR mutant. And then we compared the MROQ and the AGR mutant, and they look very, very similar. So there are some big differences, but really, when you look at it, the differences are quite small. It might be that in the mutant, the threshold wasn't reached. In the AGR mutant, it was. But if you look and zoom in, almost all the changes are identical. It's amazing. If you look at some of those highlights of changes in the MROQ mutant, they're just as you might expect. Things like surface protein we'd already shown in a block, was way up. Phenol-soluble modulins, I think that should be others. The minus, it just kind of ran over, so almost 1,000-fold down. Some of our proteases are 50-fold down. Leukocidins are way down. The SPL operon AGR control is way down, and the AGR system itself way down. The RNA3 almost 300-fold. So it just kind of makes perfect sense of AGR control. We see shared phenotypes. So here's our MROQ mutant, makes a much better biofilm than the wild type and complement, and it's just like an AGR mutant. And this kind of makes sense because we know AGR mutants make a better biofilm. Something that we'd never seen before, and I don't think anyone else has, is that at least in USA 300, the AGR mutant has a defect in terms of pigmentation. And so does our MROQ mutant, which we would expect if MROQ controls AGR. We show SIGB just for a complete pigment knockout example. And that loss of pigment also leads to a sensitivity to reactive oxygen species. So this pigment does help to resist reactive oxygen species. And there's our MROQ mutant and AGR, and they're both impaired, although SIGB being completely apigmented is way, way, way down. So shared phenotypes. And then we did some virulent studies. And so this is with a former postdoc, Ronan, who has his own lab, who's in the lab with Antonio. And so Ronan did, this is a skin abscess model where we inject the bacteria under the skin and we get an abscess. And so when we look at the abscess area for the wild type, they're kind of big and healthy, but we get a massive reduction in our MROQ mutant and even further for AGR. And then if you look at this in terms of CFUs, we get log reduction for the MROQ and multiple log for AGR. And so we say that AGR is greater than MROQ for infection. So MROQ is necessary, but not sufficient for AGR activity. There are other factors, but it does profoundly control its activity. And then we also did a septic infection. This is kind of an interesting thing where these are the kidneys of wild type infected mice. But these are the kidneys of the MROQ mutant, and this is the AGR mutant. And certainly with the AGR mutant, you see these massive abscesses forming on the kidneys that you just don't really see if you look at the wild type. You do kind of see them with MROQ. And so this is, again, phenocopying this idea. And we kind of think that this is a lot to do with the AGR mutant gets stuck in an abscess and cannot disperse because it can't produce virulence factors that would facilitate dispersal. And so the MROQ mutant's mirroring that. So how does it work? And so I guess being close to time, what I'll do is I'll wrap this story up and then I'll kind of close for you rather than I have other things I could tell you, but I don't want to be over time. So here is our AGR system, right? And so here is MROQ. We know it's a membrane protein. And so it sits in a membrane and we think it engages with one or the other of the membrane components of AGR. And so we first thought, well, let's check the quorum sensing module because we can check that because we have collaborators who do this kind of work. And so what we did was we looked at the production of the pheromone of AGR in our various different mutants. So this was Nadja Ketch at UNC. And so she's a mass spec expert who'd already figured this kind of work out. So here's purified AIP run on a mass spec, and there's the big spike of purified AIP. And if you look at staph wild type, there is an AIP. It's at the same retention time, and we can see it, and it's there, and it's all kind of well figured out by those guys for over years. In our MROQ mutant, now the peak is nowhere near as high. It's not as much. The area under the curve is a lot lower, but it is there. So mature and cyclized AIP is produced by the MROQ mutant, and it's there in the complement too, but it's not present in the AGR mutant or just in TSB. So we're thinking, okay, it doesn't influence AGRB, so it must be AGRC, right? And then we do some yeast 2 hybrid, and it does not interact at all with bacterial 2 hybrid. It interacts with AGRB after all. So it doesn't affect the ability to process the pheromone and get it outside the cell, but it certainly interacts with AGRB for some reason, not AGRD as we expected, AGRC as we expected, which was kind of a surprise. I didn't tell you this, but MROQ is itself a protease. And so here is the kind of the crude cartoon structure, and these are the proteolytic motifs of MROQ. And so we said, all right, is its protease activity required for interacting with AGRB? And the answer is yes. So here's regular MROQ and AGRB interacting. If we make this what would be a catalytically inert MROQ, it cannot interact with AGRB anymore. So for some reason, MROQ is proteolyzing AGRB to make it work. A very interesting aside, and I don't know what it means, is if you make the catalytically inert version of MROQ, not only does it not bind AGRB now, it binds ARLS, which is a 2-component system insulin kinase. I don't know what that means, but that's kind of interesting. We kind of reprogrammed it. But anyway, to stay on message for the last few slides. So MROQ engages with AGRB. And so this is the structure of our AGRB, the topology in the staph membrane. It was figured out by our good friend and colleague, Alex Horswell. And so MROQ comes from a family that should cleave the C terminus. The proteases should cleave the C terminus. And so what Stephanie did was she took away this first C-terminal part of AGRB, and we said, can the two still interact? So she truncated to this 164 residue. And it still does, right? Maybe it's not as intense, although that is the positive control, not AGRB, but it still gives us an interaction. So the loss of this first helix does not disrupt MROQ interacting with AGRB. But if you go to 141, it does. So we take away the second transmembrane helices and it goes away. And we think it's actually a lot closer to down here than it is all the way up here. But we think that the engagement of MROQ with AGRB occurs somewhere around in here. And so what we're going to do to figure that out is to use C-terminomics, not N-terminomics. So C-terminomics works in exactly the same way as N-terminomics, but you tag the C-terminus instead. And so we would find C-terminal cleavage events, use a polymer to pull and tag things out and run them through the mass spec and figure it out. And so that's our next step, is to hopefully witness AGRB cleavage by MROQ using C-terminomics. And then I think I'll try and make this my final slide. One of the things that I've definitely become clear about is that MROQ regulation is AGR regulation, right? We're pretty sure all MROQ does is control AGR. So this is some knowledge we have about AGR regulation from over the years, although only a couple of these different transcription factors have been shown to bind to the AGR operon. So we know they regulate, but we don't know that it's direct. And so what if some or more of these things actually regulate MROQ, which feeds into AGR regulation? So that's why I say, you know, our knowledge of AGR regulation might actually be upside down, because it might all be MROQ regulation through AGR. And so again, using that screen that Andy Weiss developed, Stephanie has tagged the MROQ promoter and with luciferase and put it through a bunch of different mutants. And here's some of the data from the screen that she ran recently. And we've got plates and plates more of this stuff. So we're beginning to understand things about how MROQ is controlled, which informs on how we know that about AGR is controlled. So I was going to tell you about XDRA, my new favorite regulator. I put this in here just in case I was going to run out of time, but I'll skip over because it's fun, but I guess that's for another time. And then I was also going to give a plug for, what's that?
No, it seems that somebody opened their microphone, but it's good.
Sure.
No worries, go ahead.
So yeah, so I'm going to skip through XDRA. It's really kind of fun and interesting. It's the idea of reducing cytolysis to enhance pathogenesis, but that can be for another time or a different conversation. And I was going to give one final plug for some really cool clinical strain type work we've been doing with proteases with Tajazarian and my student Jessica, but that was just a couple slides. I think it's really cool, but again, we'll have to be for another time. So I guess I'll end on what I think our model of study is, right? And so Staph aureus causes infection in every single ecological niche of the body. And so it starts by sensing the host niche that it's in. Am I in the brain or the lungs or the heart or the bones, right? It needs to know where it is. And then it changes its regulatory network. And this allows it then to fine tune the virulence factors it produces to cause the infection, the disease specific outcomes, right? And we think the proteases sit right at the heart of this, that the bug can sense the niche, change the regulation network, skip over proteases and change virulence factors to give disease specific outcomes. Or it can regulate the proteases to regulate the virulence factors to give those disease-specific outcomes. And so this is the pathway of control that we understand in my lab. And proteases kind of sit at the center of everything that we do, but we're also interested in other things around it. So with that, I am done. This is most of everyone in the lab. Brooke has now moved on, and there are a couple new students who have joined Brendan and Emily. But I want to thank, as I said, all of the people in the lab over the years who've done the work. They're the real heroes. I just narrate the work and I get the money, right? That's my job. So our funding's from NID and NIGMS. So I'll thank you all for your attention, and I'll take any questions you might have.
Great. Thank you very much, Les. This is a very interesting overview of your research for the last many years, like 10, 12 years or something, isn't it?
Yeah, I'd say that's what we've been doing since when you were in the lab. So
yeah, no, that's amazing. Thank you very much. It was very interesting. So I'm gonna open the floor for questions. Preguntas, jóvenes. So sometimes they're very shy, so I'm gonna start.
I know how it is.
So I definitely, you piqued my curiosity about the XDRA. So what kind of regulator is that one?
So it was one of those things, you know, it's in your paper where we looked at all the different regulators. An XRE type element had been discovered maybe 15 years ago as a paper from Nadine McCallum and Brigitte Bergabaki. They showed that it regulates SPA. And there've been a couple of papers over the years, but we pick it up as a protease regulator. And then we kind of look into it a bit more, and we find that it actually seems the mutant seems to be cause more disease, not less. So in 2 different models, it has enhanced virulence. And at the same time, it seems to have reduced cytolytic capacity. So it produces less alpha toxin and things like that would lyse host cells. And as a consequence, we think it makes it more stealthy and allows it to survive better in the host. So it's kind of a backwards way of thinking about it. But this has happened a couple of times recently. There have been maybe 3 mutations described in the literature in the last 5 or 10 years that when they occur, you actually get better virulence, which is almost upside down to how we think. But I think we're beginning to realize that staph being stealthy is sometimes to its advantage.
Got it. So now that you mentioned this, that paper we wrote a while ago, isn't that similar to what some of those phage genes regulate?
Yeah, so it's XRE means xenobiotic regulatory element, I think, and the classic examples are phage encoded. This one is not. This is core genome. But there are so many phage regulators that we pull that are from your paper that we get phenotypes for. So even though they're on a phage, they do something to the bug, and we know that, right? Think about the STX regulation in E. coli, right, and DTXR and the like. So yeah, it is core genome encoded, but it is related to the phage guys.
Right. So there's a question here by Yesenia Picasso. Could it be possible to use these findings in a way that help us to better face antibiotic resistance? And if so, how?
Yeah, right. So all of us who work on these kind of genetic regulatory pathways, right? We say we're going to study this to find a new pathway to target for antibiotics. The proteases, at least, that causes a problem, right? Because if we start to interfere with protease activity, we actually make the bug more virulent, not less. So I always struggle when I'm writing this stuff in grants, because I can't say, well, I'm going to use it to figure out a new pathway for antibiotic treatment, because I might create a super killer in the process. So proteases themselves are the target of many different drug options and trials. They're 5%, I think, of the human genome is a protease. So I think that we could selectively maybe enhance or inhibit the activity of protease if we understood the nuance of the system. So maybe something like that, or its regulation. Certainly with our MROQ work, right? If you impair MROQ, then AGR goes away, and AGR controls most virulence, although you still pull AGR mutants from dead people, right? It causes infection and kills people. So anti-virulence type strategies are tricky. They're certainly attractive. They've got a lot of attention. Not many have worked. And I'm, when I teach undergrads, I say never, right? The minute you say something can or can't happen, the opposite occurs, because that's science. So I don't know, honestly, if that will be the case. I think for us, it's more one of those things of us providing the basic science and knowledge as a foundation that someone else maybe is probably more capable than us to figure out how to make it a treatment. But yeah, antivirulence is certainly something that gets a lot of people's attention. I just don't know how we do it. It'd have to be very nuanced.
True. So there's another question by Guy Aragon. What could be the advantages for the bacteria to evolve these circuits involving RNAs and proteins?
Right. So, I mean, if you think about evolving multiple circuits, which is what we see with bacteria, right? Staph, I think from Antonio's paper, it's something like 140 regulators that we figured out. And then in the other work by a postdoc that was with Antonio at the time, Ronan, we found another 300 regulatory RNAs, right? So that's a lot of options. And then if you think about what we've been showing with the proteases as post-translational regulation, I mean, and I think that's the beauty of bacteria, right? They're supposed to be simple, these kind of single cell, one bag of biochemistry, and they're supposed to be way lower on the kind of intellectual chain than eukaryotes, but they have no compartmentation. They have all this complexity, and they still are super successful. And so that's the thing about bacteria. They're just very efficient. And so part of that efficiency is just everything is controlled in the cell in just an exquisite way that's so tightly regulated. And that's, when I teach undergrads and grads, whenever I ask a question, I say the answer is always regulation, right? Any question I ever ask, the answer is regulation. because that's what bacteria do so well in my mind, right? Just one bacteria that, a small genome for staph has got 2,800 genes. So that's, 2.8 megabases. E. coli is 4,400 genes or more. So not many genes, but still thousands of regulatory nodes. That's the ultimate level of control, right? If you precisely know everything that's occurring in the cell and how to fine tune it, that's what makes you just that little bit of more efficient than the competition. And that little bit of efficiency over time leads to dominance of a population. And when the bug dominates the population, it eats all the food, it wins. And that's what all they're trying to do.
True. Yeah, so Raimundo Ramos wrote, it's not a question, but I'm really amazed about the research your team has been doing. It seems quite complex, and to be honest, I'm still trying to digest it. Thank you for sharing your research with us. That's it. Thanks, Raimundo.
That is Raimundo. That's very kind of you to say so.
I have sorry. And I would like to talk about it real quickly. I didn't type anything up. But...
Go ahead. No, it's you went up in your microphone. I forgot to say you went up.
Yeah, I wasn't sure if typing or not, because it's just, number one, I really enjoyed the presentation of the stuff that's happened to the lab since I've been there. Andy Frey's work is amazing. I was like writing down questions about continuation on Brittany's work, and then I was like, oh, Andy already did it. But then I was like, oh, it's just serum. What about the organs? And then boom, organ work. So it seems like everything was following up on what I was going to ask. But I do, as in my own work, I've been becoming more and more interested in the host's response to infections, particularly sepsis. And that kind of goes into the antivirulence realm. But I was wondering if you were thinking of collaborating or doing any work to see the effects of your mutants or all of Brittany's mutants on the host, like the cytokine secretion or macrophage recruitment, or perhaps sepsis of the infection. Because I know gram-negative sepsis is a huge killer, and they still can't seem to get their hands around therapeutic treatment for sepsis. So I was wondering if you were planning on doing any of that in the future.
No.
Thank you.
I'm not.
Come on.
No, the reason is, right, those are all wonderfully good questions, right? And they're important questions. But I mean, anyone that knows anything about me is I hate immunology, right? I'm pathogen first, host.
Yeah.
No, I'm not even a second, right? So those are good questions. An immunologist, if we had one in the department, I would love to work with. But there's just so many bacterial questions that I want to know, right? Because why does it kill them? Why did you get hypervirulence? In 24 hours, you get this toxin storm and they're dead, right? And someone once said to me, oh, that's going to be a MyD88 response. I don't know what that means, but apparently it's the answer, right? So I'd love to know why that's occurring. But one of the things that I've learned, perhaps without, and it's, it's a cop out on an answer, but it's also true, is there's only so much you can achieve, right? There's so much science you want to do, and then there's only so much you can do. And what's within your control or your ability to control and also get the government to pay for. So even though I would love to do those things, in terms of looking at it from the host side, that would be something I would collaborate with if at all. And then your question about, sepsis, right? How is it we still don't have a sepsis antibiotic, right? That's the thing that whoever's got that has made a big difference. So for staph and E. coli are probably the two leading sepsis problems. So you get staph sepsis with, maybe 12 hours, you're dead. So,
I thought I saw a little when you were clicking through the XDR thing real fast. I was like, oh, I think I saw the word sepsis in there.
Well, we use the sepsis infection, right? So for in a septic infection, we'll do those studies. But and so that massive killing of the protease mutants is in a septic infection.
Yeah.
Why is it occurring, right? What is it? And it's a host response that's doing it, right? It's an overreaction of the host to all these toxins that's giving you this outcome, particularly that 0964 that we've studied. You take that away and it goes away, right? We've been working with 0964 for the last year to try and figure out how it does that in the host. So yeah, my first answer was still my real answer. No, but you're more than welcome to do it if you want.
Yeah. I like to tread into the deep waters. It's a little too far.
Please do. But piece of advice for you when you're running your own lab: definitely start out doing this, but you'll realize that at some point you need to do this, because you're already going to have students like you who just kind of run off and do whatever they want anyway. So
Ryan's been trying to reel in my TLR4 excitement for quite some time now.
He's fighting a losing battle there.
Yeah. Thank you so much.
Thank you, Renee. Nice seeing you.
Nice seeing you too.
Juan, do you have a question? Juan and then Guy. Juan?
Yes, thank you. it is quite interesting, this possible role of MROQ over AGR. As you say, it may change the whole view of how it works, no? So I'm interested in it. You showed the interaction of MROQ with AGRB, but you also said that MROQ may have proteolytic activity. Have you shown proteolytic activity on AGRB?
Not yet. So we know that when you stop MROQ being a protease, it doesn't regulate AGR anymore. So we did it two ways. So in the MROQ mutant, we complement back with a deficient MROQ. So you take away the protease residues and it never complements in the assays. I didn't show them for time, but you can't get it to complement. Also in that bacterial-2 hybrid showing engagement, you take away the protease residue, you lose engagement. Right, these are indirect, so we don't for sure know that that's happening, but it kind of makes sense. You know, my training is in not just staph, but protease biology. You know, a catalytic pocket is perfectly designed in nature to accommodate a substrate, right? Sit, and then you get cleavage. And so the minute you change the catalytic pocket, right, you distort that, which is what our mutations might have done, then you can't accommodate and dock anymore. So I believe that that fits in terms of the binding and the loss of the residues means we should see proteolysis. And that's why we want to do the C-termonomic study to figure out the proteolysis. It may still be an indirect effect, even though we show binding. I don't know the answer to that. And that's the next step of that project, is the C-termonomic study. And so people in the lab hear the phrase C-termonomics a lot. They're not all lining up to do the C-termonomic experiments. Andy, the N-termonomics guy, is actually heading back home to England soon. So people in the lab talk about wanting to do C-termonomics. Hopefully someone will do it soon and we'll figure out exactly, visualize that cleavage of AGRB from its C-terminus. And then we can say, oh, it just needs to nibble off a bit from the end, and somehow that activates AGRB to be more effective at what it does. Because we know that it doesn't have a role in producing AGRD, because we can still see it fully formed outside the cell.
Okay. And the mutation experiments on AGRB, does they involve the phosphorylation site? Or
sorry,
the NA site?
The little black marks, is that what you were referring to? So those are actually, I didn't explain it, but those are catalytic residues too, right? Because AGRB is a protease. So those are the residues in AGRB that we know are important for its proteolysis of AGRD. Same thing for MROQ. That was annotated for a grant I just wrote recently. And that was nicely in the figure legend, but not explained in the presentation. My apologies.
Okay, thank you. Thank you.
Of course, thank you. I like having the rest of the lab here. I could just make them answer the questions instead of me.
I was hoping you wouldn't do that.
Yeah, I could just say any N-termonomics questions. Andy Frey, you're here, and MROQ. Stephanie, her surname is Mariquin, so MROQ is kind of named after her name, Mariquin.
I was the one who is going to ask the other question.
Please.
Thank you for explaining all of this. I'm still an undergraduate student, and I don't get all of the things that you explained. I don't want to sound rude that I didn't pay attention or anything like that. But I already took immunology, and I get some things. I'm currently taking microbiology, but the question is, what was really like the main purpose of the experiment? I know that you explain it, but I don't get it full of the idea.
It's okay. You don't need to apologize, right? So what I just summarized was a decade's worth of work by 8 people, myself included, right? In 55 minutes. So most people with PhDs aren't going to understand it, or people in my own lab aren't going to understand it all, right? I mean, that's one of the beauties of running a lab, is that you get to have this empire of science you do, and you might be the only person in the world that understands it, and maybe not even then sometimes. So I think it comes back to that initial thing that I said, was that 20 some years ago, I walked into my PhD with my PhD advisor, and he said, here are the secreted proteases of staph. I think they're interesting. And then when I started my own lab, we still didn't know if they were or not. We thought maybe they're important for infection, maybe they're not, right? I'd published contradictory papers that said yes, that said no, said maybe. So that was that's my initial question. And often the best ones are the simplest ones, right? My question was just, what do they do? Are they important? Can I just show they are or they are not important so I can get on with my life? Because I keep thinking about these things. So that was the initial question. If we just take them all away, take every one of them away and then see what happens. And it was just kind of one of those fun experiments. And over here, the NIH has a grant mechanism that's high risk, high reward, right? You can propose to do something wild, and if it pays off, then it would be really cool. And that's exactly what this was. I wrote one of those grants and said, I don't know if anything's going to happen, but it might. So we took all the genes away and then we did some tests and we found out, oh, actually, we opened a can of worms, right? It's like Pandora's box. Everything just spiraled from there. And so now we figure out, yeah, they are actually important. And so now we begin to unravel individually. Of these 10, well, which ones do this and which ones do that? And so, that one question, that one grant, those experiments Antonio did on that one paper, that's my next 25 years, right? It's figuring out all the different subtleties and nuances of the system. But the initial question was a big one. Are they important? Yes or no?
Okay, I get it. Yeah, no, thank you for the explanation.
Of course, no problem. Thanks for paying attention and listening. That's great.
Thank you. Thank you. And this is very, very exciting. What I like about, all what you described is very interesting, but then you say it's so easy, but for all of you guys who are going into this field, moving from ex vivo or in vivo, going to the animal, it's really hard, and you show it really nicely. I think, well, that's another one of your strengths in your lab. I mean, you move easily into checking all these mutants and hypotheses into the animal model. So that's really cool.
That's any of you guys that have worked for me, it's always, hey, we should do this, and then you guys go figure out how to do it, right? It's not me. It's just I know we need to do it.
But
people just figure it out.
Yeah, I know. But now that I'm in academia, I mean, I try to get things done.
Yeah, I understand.
You gotta go through the ethics committee and proving that you did the actual thing, and then getting the money to do the mice work. No, it's not that easy. I mean, when you're an undergrad, a grad, or a postdoc, you really live in a nice world. And somebody gives you the money to work with, and then somebody lets you do that, and then now when you are in the actual world, is that shoot, that was it's a lot of work to do. But
I mean, that's what we do, right? It's we as a professor, and it's almost like what we do is a mystery that people don't really know what it is we do. That people in the lab know that I'm not around a lot, and I always seem to be busy or annoyed, and they're like, what? The guy just runs a lab and teaches a bit. What's he so? Right? It's all those other things are behind the scenes, right? It's exactly like you say, all this IACUC stuff with the writing of those protocols, keeping on top of the compliance and making sure that you've got all the biosafety in line. And so just the paperwork for research. And then, as you say, you're going to have the money to pay for the research, and then you're going to have the contact over here or there to do it. So, our role is facilitation. We do all this behind the scenes kind of puppet work, so the stage becomes set for the trainees to just go do. And hopefully it becomes easy to go do because we took away the barriers. But as you said, our part, particularly universities, love red tape and paperwork and bureaucracy. And so, it can be complex.
There's another question. Well, there was another question from Adriana, but I think that you already answered it about MDROQ and AGRB. So I think, but she thanks you for your presentation. And then Yesenia Hasso, what are the main factors involved in determining the difference you found in infection in different tissues in vivo test? I guess that would be a summary.
Right. So I guess we're talking about the different protease mutants. So for those at least, that looking for the hypervirulence, if that's what we're talking about, that one's fairly straightforward because even though that model can be used for so many different things, our main outcome there was just survival. So we're just seeing if the mice survive the infection over a course of a couple of days or not, the hypervirulent killer phenotype we'd engendered. So really, we're actually using a very crude approach to how things are going because we just got one easy readout. And then some of our other more detailed studies, right, we'll do a septic infection. You can disseminate to all 6 organs. And then, yeah, there are, as you know, Renee pointed out, there are many host factors along the way that are roadblocks to prevent the infection occurring. And staff's primary success is from evading the immune system through various different mechanisms too. And so I think it's just what staff does. And I've already said that I'm biased, but it's got so many diverse virulence factors for such a small genome that there are multiple different primary effectors that it can use to get through the host and survive in each of the different niches. And then, you know, the host is using its own defense systems like those antimicrobial peptides, the complement system, cell-mediated immunity. And staff, what it does is a lot of bugs will hide. They'll know that the immune system exists and they'll hide from it. Staff goes looking for that stuff. It picks a fight. It says, I'm here and I can take you down. Because it's just so well armed. So, and that's what's kind of fun about it. Is you're really studying warfare when you're looking at this kind of stuff between the bug and the host. And so, and then for us, in terms of figuring out what we think are the most important factors for the progression of the infection, that's that kind of next generation proteomics work that Andy's been doing that we're hoping to be able to study over time, just in a wild type, how infection occurs, and then looking at different mutants and how that infects infection, then different kinds of infection, or then in one infection in different parts of the host. So yeah, that's actually more than my entire next career's worth of work to do. Now I'm thinking about it. So there's way too much to do.
Yeah. So I have 2 other questions from technical questions, but I think Juan has another one. Juan?
Yes, thank you. Speaking on infection models, in vivo models are quite difficult. What do you think about cell culture infection models?
Yeah, they're very good, and we've certainly used them quite a lot. It's actually funny. It's actually easier for us to do a mouse study than it is to do cell culture, just because, right? We have great animal facilities at USF. We get the mice. We very well experienced. We've got good help. It's kind of with the cell culture models, which we have done, and certainly when Antonio was around, we did a lot of those too, right? People who come with those experience and know how to do those things, which Ronan and Antonio did. So there are several people in the lab who do those too. But yeah, depending on the trainee, there's often more willingness to go into a mouse because it's what they know versus cell culture. But we've done macrophage and neutrophil survival studies, and you know, over a course of time, and we've collaborated with other people looking at kind of almost Trojan horse type survival of staff. So yeah, the cell culture models are really very, very powerful.
Okay, thanks.
So now, Montserrat, you were raising your hand. Montserrat? I think okay. So let's, which, do you still use your iTorrent, or you changed it already?
We got rid of it in the end. So we used it up till about maybe 2 or 3 years ago. And it was, the Iron Torrent was great. We got a lot of good stuff out of it. It was like, you know, like having an old car that would break down a lot and you love it, but you hate it. And eventually the genomics facility on campus was buying all new Illumina stuff. And they said, oh, if you will trade in, so Illumina would take my Iron Torrent as part of a trade-in to buy their stuff. And it was kind of a deal where if I did that, then I could kind of get an inside track on using the new Illumina genomics facility. So they bought a MySeq, a NextSeq. They've got single cell stuff over there. And so we, in the last 2 or 3 years, have done all Illumina. But you know, I think we published our last, well, maybe actually we're still actually about to publish some PGM data from genome sequencing. Sarah's got a paper in review that's got some of that in it. But
cool.
Yeah, it was good to us while it lasted, but we were not sorry to see it go.
Okay. And the other one, your 2 hybrid system, which one is that? Because that really, I'm really interested in that because it gives a color, so that's really cool.
Yeah, it is. So it's the split adenylate cyclase system, and so I think is it Borrelia or Burkholderia? It's one of them. They split the adenylate cyclase, and so you then tag it. And then if they come into contact, the adenylate cyclase works. So you get cyclic AMP, which hits the lac operon, and away you go.
Yeah, they use Bordetella pertussis.
Bordetella, there you go.
So you got it working, because I remember it's a little bit of pain, isn't it? Because I remember I saw that one. I was no, I didn't want to use it, but it works really nice when it does.
So this is the trick, right? And yeah, correct. Because you did all that yeast 2 hybrid in my lab too, which we still have that data, and I still think about that stuff from time to time. And Christina in the lab actually ran with your stuff and got more hits. But so the bacterial 2 hybrid thing I brought with me from Missouri, and it never really worked. But our neighboring lab is a cell division lab, and they use it all the time, and it seems to work. And so Stephanie, whose work is all the MROQ stuff, she got a supplemental grant from the NIH for her work, and she got several thousand a year were her supply money. And so when we got that, I said, you can spend it on what you want. And so I'd heard from other people that if you get the bacterial 2 hybrid plasmids directly from the source, which is Pasteur Institute in France, they work. Somehow, if you get them from someone else, they don't work. And we went through a bunch of paperwork, but we got them from Pasteur. It was not too expensive, and they work. It really worked. So Stephanie's made libraries of these things, amazing stuff, and it works. It's been great.
Good to know. I'm gonna try to do it, but like I told you before, getting things to Mexico sometimes it's a hustle. It's a pain in the butt.
These are just two E. coli plasmids, so you might be all right.
Yeah, I hope so too. So if you try to get them from Adgene, let's say, they don't work. You gotta get them from the Pasteur.
I forget. Stephanie's on the call. She can remind me. What did we do?
Sorry, I'm eating. So we actually ended up buying them from Euromedics. They're like the only ones that sell bacterial 2 hybrid kits right now. Euromedics.
Okay.
I tried finding anyone else that would do it, and they're apparently really the only system that's left for you to buy. And they do ship from France, so it's a little complicated, but I've sent a bit of strains out, and I think to Ronan, right? Yeah, it was to Ronan, and he's had luck with them, I think. So maybe it might be easier for me to ship things to you rather than from France. I'm not sure.
Great. We can send you some tortillas if you find them.
Oh, please. De maíz, por favor.
Oh, yeah, corn, of course.
Of course.
Yeah, then it's a deal.
Yeah.
Thank you, Stephanie. Well,
yeah.
Thank you very much, Les. I really appreciate your time, your answering your questions and all that. I just want to share with you. I'm gonna send this to you by email because there's no other way. But yeah, this is something we prepare. I hope to see you soon so we can share a beer or a bottle of wine, whatever.
Absolutely.
Hopefully this pandemic will let us come and visit each other. I really hope to go. I enjoy my time in Tampa in your lab, and I learned a lot, like I told you before, learn a lot of things, and it's very nice to have you here. So thank you. I really appreciate you sharing your research and your time with us.
Thank you. And it's, these kind of visits are, particularly when before the pandemic, I used to do these a lot, but these ones particularly with people and people you care about and you've worked with before, these are ones that kind of really are the most fun and the most enjoyable. So it's so much fun, and I really appreciate you asking me to come talk.
No, thank you. I also did it because I really want to start over again the Staphylococcus, and like I told you, hopefully Juan's project, Adriana's, we'll go ahead and start getting some more data from mutants and things like that. But we're doing steps, and it's going to be hard to get your pace, but we'll try.
We're happy to help in any way we can with that, honestly.
Thank you very much. And I'm not sure, guys, you want to open your camera and maybe we can take a picture. It will be nice to be in person, but
yeah, sure.
Okay. Thank you. So who else wants to open their camera? Okay. Nice to see you. So many students, undergrads and grads. So I think that's as many as we're gonna get. Okay. Here we go. 1, 2, 3. Okay. Well, again, thank you very much, Les. Thank you. It was a pleasure to talk to you. Please give my best to all the members in your lab, to Jess and Jack, and hope to see you soon.
Absolutely. Thank you all very much for your attention. It's been great. Thank you.
Thank you. Muchas gracias a
Bye.
todos.
Thank you very
Gracias
much.
a todos. Dejé la liga para los que necesiten constancia. Y muchas gracias a todos. Thank you again, Les. Thank you.
Of course,
Thanks,
thank you.
Stephanie.
Bye.
Bye-bye.